Okay, thank you, Roberto, for this kind introduction, and thank you, everyone in the audience, for joining in today. And today, I'd like to tell you the story of a journey, a scientific journey that started some 15 years ago, around 2004, when I was finishing my postdoc at ECT Star. At the time, I was working entirely in the framework of theoretical quantum chromodynamics. Therefore, I was interested in problems related to the quantum fluctuations of the vacuum and the implication on the physics of atoms, something completely unrelated to the biological aspects and topics that I will touch on today. However, while I was working in QCD, I had a very inspiring discussion with Henri Orland from S.R.L, who later became a collaborator of mine for many, many years on these topics. And during this discussion, I became aware of a very fascinating and inspiring analogy that can be found between the physics of hadrons and quarks and gluons on the one hand, and the physics of biopolymers such as proteins on the other hand. You know, hadrons can only be found in very specific combinations. Quarks and gluon can only arrange themselves to form white objects, or color singlet, more technically speaking, object, which provides just a tiny little fraction of the possible combination of quantum numbers. But if you heat up a hadron sufficiently high, then it deconfines and releases all the possible color configuration you can think of. That's the famous deconfinement phase transition of QCD. I was also interested at the time. Well, proteins that are the biological workers of the cell, they are found in one specific conformation in which they perform their biological function. And if you heat them enough, you can melt this conformation just as much as hadrons, releasing a very large amount of configurations when the protein is so-called denaturated. So there are many other analogies like this, and sort of they're kind of reminiscent of the fact that both systems have like a large fluctuation, strong correlation, or whatnot. But at the time I began, it could be possible to exploit some of the mathematical methods that physicists have developed to study strong interactions, for instance, of fields. So very advanced, very sophisticated methods, and maybe adapt them to study proteins, and therefore, in some dream, at the time at least, being able to envision or help out in envisioning new therapeutic strategies and be biomedically relevant. So going from QCD to drugs, so to say. And the journey I'd like to tell you about is how over the last 12 years or so, we sort of tried to sketch a line connecting these two parts of this plot, from the top, from the board, to the bench downstairs. So that's what I'm gonna do. And I'm gonna straight go to the point of the main challenge. Suppose you want to apply the physicist approach to biology, meaning the reductionist approach. What I mean by that is, rather than look at biology from a systemic point of view, say top down, you look at it from a bottom up point of view. You try to look at the molecule and try to understand from an atomistic level what's going on, and then use this information to, say, gain some information about biological problems. Well, if you want to do that, you have to solve typically a million or a few million coupled Newton's type differential equations. That's a large number, of course, because you have all the atoms in the molecule and all the atoms in the solvent around the atom. But not only that, you have to do that for an extremely long time, because typically biology works through rare transition. Let me elaborate a little bit more quantitatively what I mean by rare transition. And I would take as a prototype the protein folding relationship, the protein folding reaction, for reasons that will become clear later in the talk. You know, proteins are these biopolymers produced as strings by the ribosome, which reads the information in the messenger RNA. And after they are produced, they sort of self-assemble themselves to reach that unique native configuration. But in order to do that, they have to cross a free energy barrier, which has both an entropic and an energetic contribution. And anytime you have a barrier and you are in a thermal activated environment, meaning you have a heat bath around you, well, you can show, it's a well-known result since Kramer's in the 40s, that you wait an exponentially long time before the right sequence of fluctuation event. You can picture this as, being the bottom of the valley and waited from random fluctuation to kick you on the same direction a sufficient amount of time for you to reach the top of the mountain, and then you can roll down on the other side. And of course, it's like flipping the coin an enormous amount of time and getting always head before something can happen. As a result of which, if you look at a plot, you have an order parameter, say, for protein folding. Typically, they look like this. You spend an enormous amount of time in the reactant, and then in a very short time, you make a transition to the product. This decoupling of time scales between the time you have to wait to see what you want to see is the reason why molecular dynamics simulations cannot be applied to this problem, essentially. Well, even using the most powerful supercomputer on Earth, which is designed to do only molecular dynamics, you can typically scratch the millisecond time scale of simulated time for very short, almost synthetic proteins, meaning proteins whose length is so much shorter than the typical length of biologically relevant proteins, and folding time a couple of orders of magnitude at least smaller than the typical folding time. But you can actually scratch the very, very tail of the typical proteins in the proteasome. Otherwise, most of the time, what you're simulating is just fluctuations in the reactant state. You look at the protein just moving around without folding. I was referring to this supercomputer that in 2010 was able to fold by molecular dynamics a set of, I think it's 12 mini protein. These are engineered protein, basically fragments of proteins that can fold in sub-millisecond time scales, but they were used as a proof of principle that molecular dynamics can fold proteins. But from a biological point of view, they are completely useless, and typical proteins can fold in tens of seconds, minutes even, and several seconds, say, would say, is the standard number figure. So you see, there's a big difference between the simulation zone, let me call it, and the biozone in terms of time intervals. This is the reason why over the last 20 years, many groups around the world have tried to develop advanced methods to tackle rare event problems in macromolecular dynamics. And typically, all these methods, with some caveats here and there, share in common one feature. You provide a calculation, more information, and you hope that with this more information, you can shorten the simulation time. And there's never a free lunch, of course. There's perhaps a couple of exceptions, but I won't go into that level of detail. But the point that I want to make here is that in spite of the fact that these are very elaborated, very smart methods, none of them is sufficiently efficient to be able to, say, fold a protein of 300 amino acids folding in 20 seconds. That's a typical time for, I don't know, biologically relevant proteins. And that's where we sort of came in. Remember, the idea was to try to use methods that originally were developed in the context of theoretical physics, try to help in bridging this time gap. And this is a work that at the beginning was done in collaboration with Henri Orlando in Saclay and also with Francesco Pederiva here at Trento. And the starting observation came from noticing that thermal activation, which is the process I just discussed about, you are in a classical environment, but in contact with thermal fluctuations. So every so often, you can actually cross a barrier. Displays some analogies with quantum tunneling. You are on the left-hand side of a barrier, and because of quantum fluctuation, your wave function extends to the right side of the barrier, and you can actually tunnel across the barrier, right? And it so happens that there is a mathematical formalism that can be used for both types of problems, and it's the path integral formalism. Downstairs is the Euclidean path integral, Feynman path integral. You don't need to understand it, it's just to see that it exists, which is the basis of what is called the instanton theory for tunneling transition. And that was the subject of my PhD and my postdoc work. So that's my angle into this problem. I knew quite a lot about this theory. On the top is the probability of performing transition expressed as a sum of all possible paths through which you can perform a transition. each one with its own probability amplitude. So this straight analogy was the gate into trying to use tools that were used in QCD to study tunneling transition in the vacuum, and that's wall research field, and that's what I was mostly doing at the time, and trying to adapt them to study thermally activation transition of biomolecules. Now, what is the advantage of using the path integral formalism with respect to doing molecular dynamics? Because you see, if I go back, you see that in the path integral formalism, I have the probability to go from one point to another, and these 2 points are given. That means that I can provide this information that is experimentally available. For instance, I know that unfolded proteins are more or less like random coils, and I know that the folded protein, the native protein, have a specific structure that is resolved by X-ray or NMR experiments. So I can provide this as input. This is the additional information I was mentioning before. But now the path integral focuses directly only on the transition going from these 2 configurations. So in other words, you're focusing directly on the reactive part of the dynamics, and you completely neglect everything that has to do with the waiting time. So basically, you only focus on the shaded area in this plot, and you completely skip and you avoid wasting computational time in the waiting time zone. And if you work out the physical chemistry of this, you can realize that the typical time it takes for a system to cross a barrier along a productive trajectory is doubly logarithmically shorter than the typical waiting time. So you have a doubly exponential gain, if you want, in computational time if you restrict to the reactive segment of the dynamics. Now, even if you do that, the path integral you have to solve is so high dimensional, million and million dimensional, 10 to the 12 dimensional typically, you cannot hope to solve it by Monte Carlo. There's no way you can solve it by Monte Carlo or by any of the standard method of computational physics. You have to invent some. And that's what we did for the last 12, oh my God, it's already 15 years, fine, with a series of P.R.L. and PNAS papers right here, with Henri Hollande and Francisco Pederiva, and then they were basically a copy of this instanton theory I was referring to, adapted for molecular dynamics. But then we moved on and developed more advanced and more powerful algorithm. And I don't have the time to tell you how they work. I'll be happy to discuss them in the discussion session. But let me just say that all they have in common is they're all variational approaches. And in some sense, you introduce some additional approximation with respect to the standard Monte Carlo. It's more like a variation of Monte Carlo, or in some flavor, a combination of variational and mean field. So you have to introduce additional approximation, but these are approximations the nuclear theorists have known for 60 years or more. These are well-tested approximations in theoretical physics. And so I think this knowledge, we all more or less know when they work and when they don't. So it was kind of natural, although they are very different from their formulation in nuclear theory, the general spirit is similar. And so in some sense, this is part of the arsenal of ideas of the mean square theoretical physicist in many-body theory. The first thing we ever did with that was to check whether we can get reliable results compared to the standard molecular dynamics simulations. And then, so what we did, we collected a sample of those famous 12 proteins that were folded by MD, and their data are available for download. And we simulated the same identical folding processes. And what these plots show, without going into any detail, is that the overlap of the curve simply means that the mechanism for folding you get from our method was statistically indistinguishable from the method for the mechanism of folding you were getting from plain molecular dynamics, given the same system, given the same model for the forces. So basically, we were getting indistinguishable results from molecular dynamics simulation as far as the folding mechanism is concerned. Okay, but like I said, we can extend much further. Basically, with these methods, you can go as long as hours in terms of simulated nominal time. You can look at processes that can occur within hours or tens of minutes, as you can see some examples here. So in some sense, after you validated the method on the zone where the simulations are available, for the first time, we could really explore the biozone, I call it. The zone in time windows and in number of atoms that is biologically relevant for, say, protein folding or similarly complex transitions. Okay, so that sort of introduced the idea that we had, the angle we had into the problem, and it was nice to see that you have an agreement with the molecular dynamic simulations. But as a physicist, I would not believe anything that doesn't have a comparison with a real experiment, because it's good to find, reassuring to find comparison with simulation. But we all know that ultimately are the experimentalists who decide what's true and what's not. So we try to build up a direct comparison with experiment. And here we face a challenge. One of the problems, according to my opinion, at least of biophysics, is that it's very difficult to perform a direct comparison between experiment and theory. Most of the time, you are stuck with qualitative comparison. You get some qualitative information from biophysical experiments, which you match against qualitative information you get from simulation. For example, you know, this part of the protein gets exposed to the solvent longer than this part of the protein. Oh, fine. Also in my simulation, that's what happens. But as physicists, we all know that until you can put a point and a line, and the line is the prediction, then you really don't understand things thoroughly. So the challenge is how to try to do that in the cleanest possible way. And a clean way is one that doesn't involve putting mechanical strain on the protein or changing the chemical structure of the protein. And as physicists, we know that, you know, the first thing we think of is scattering, right? Inelastic scattering, typically. And for instance, a standard technique, I mean, translating from, say, biophysical to physical language, what typically experimentals that can be done is time-resolved spectroscopy experiment, which from the point of view of physicists look like this. You have a protein that begins to fold, reaches some state, absorbs a photon, gets an electronic excitation, emits the photon, continues to fold, and ultimately reaches the native state that is on the right, and in my screen is covered by the numbers of attendees in this talk, but there's a native structure. Now, if you collect all this radiation that is being emitted at different times across the reaction, you basically are literally seeing the folding process with the eyes of emitted light. And of course, you have to deconvolve this information in order to see what's going on with the protein. Now, the challenge that is that in order to match this information with structural information and get some insight in what is going on and compare with the experiment, you need theory to predict that radiation, right? Just as much as you would do in LHC, right? You predict the outgoing beams. And in order to do that, you need a theory for non-equilibrium quantum dynamics of electronic excitation propagating in a conformationally evolving protein. And that's an awful problem, as you can think of, because already quantum dynamics is a challenging problem. This is a non-equilibrium open quantum system with a background of a protein that is moving. We spent several years in trying to develop the mathematical formalism to address the problem. And we came up with a theory that we called molecular quantum field theory. This was done in collaboration with a PhD student of mine, Elia Schneider. And I will not go into these details of this formula, but there is an interesting reason. So basically, the idea underneath this is that we were able to map this problem, that is a non-relativistic problem, but out of equilibrium in an open environment, into a virtual, so completely mathematical mapping. There's no physical interpretation. Equivalent problem that is a relativistic quantum field theory in vacuum. So we were trading the complicancies of non-equilibrium non-relativistic dynamics with the complicancies of relativistic quantum field theory in vacuum. But that is a theory that, you know, nuclear theorists and high energy physicists have a lot of tools to deal with. So this duality, I can elaborate that in the discussion session, was very useful. And it turns out that you can formulate the problem that I just drew before as a diagram in terms of this path integral, which has fields with matter and virtual antimatter and configurations and Lagrangians. And if you, those who have a background in high energy physics, they will recognize that there's a structure very similar to that of QCD or any gauge theory. And once you have done this mapping, then we spent a few years in, adapting the tools that we know from field theory to this problem and being able really to compute the time evolution of the density matrix in an open quantum system associated with a molecule and the nuclei as they propagate, and being able to make predictions for systems where you absorb light and emit light. In some cases, you can even simplify grossly things and do some more sort of a phenomenological approach. In other problems, we were more rigorous. And I don't go into the details. I'm going to show a couple of plots. The calculation on the right was done entirely rigorously in this framework of this quantum field theory, and its comparison with some linear absorption spectra for some photosynthetic spectrum against the experiment. And on the left-hand side is some simplified calculations in which we tested if we were able to accurately predict the protein folding pathways, or more specifically, if we were able to predict the sequence of intermediate states that a large protein takes while it's trying to reach the final state. And the outcome of this study is on the left, and on many systems, on different systems actually, was that we were surprisingly well able to predict and reproduce experimental results on time-resolved spectroscopy for protein intermediates. In other words, our predictions for the folding intermediates was actually extremely accurate. And beyond any hope sincerely, we could think it was reasonable hope. So in other words, we had almost quantitative agreement with the experiment. Of course, there is a little bit of disagreement on the left, but there's a number of assumptions about the system that is huge. And most importantly, if you just tightly change the configuration of the protein, you will get a completely different signal. Meaning that really our calculations were really quite accurate at that point. So with this in mind, we felt we were well equipped to move into the biology territory and try to address biologically relevant problems. And this is, this is a, okay, fine. This is a slide that just summarizes some started to, and of course I will go to none of them in specific detail. But the only point that I want to say that during this work in biology, a major event along this journey occurred, and was to team up with Emiliano Biasini's lab here at Icibio, and together with his PhD student Giovanni Spagnoli, who's basically a senior member of the lab in spite of his young age. And that was a game changer because for some reason we were able to find problems where we could really match biological competence and biotechnological competence with computational problems we could solve. And that sort of boosted up our interest, and we melted up our groups. Now we have a group where Giovanni wakes up, does simulations in the afternoon, synthesizes a molecule, and in the evening goes to do some NMR experiment. So really it's an interdisciplinary group. And we started looking at some biologically relevant questions, such as how amyloidogenic proteins start to form fibrils or something like this. And along this sequence of studies, Emiliano and I came up with an idea which finally led from quarks to drugs. An idea to use all this machinery we have for studying protein folding pathways to design drugs. So finally, to bridge the gap towards biomedical applications. And let me tell you a little bit about this idea. In order to do that, I have to say something that for some of you may look trivial, for some other maybe not. Most of biological functions in cells are carried out by proteins. They help transcripting genes, they do quality control, they transport, they do whatever you need to do is done by proteins. So this is the reason why most of medicinal chemistry and pharmacology is based on interfering or inhibiting with the biological functions of proteins. Some biological function is harmful. You recognize what proteins are responsible for that biological functions, and you try to interfere with the biological functions of those proteins. This is the rational approach to pharmacology. There's also a purely serendipity type of approach to pharmacology. But of course, that's not a place for science. That's a place for big facilities and make serendipity statistically significant. So as far as the rational approach to pharmacology, proteins are the game, are the object to look at. And now in this slide, you see several ways that people have come up with in order to interfere with the biological function. So on the top of this slide, I sort of synthesized for you what is called as fundamental dogma of molecular biology. Namely, you have genetic information stored in the DNA, gets transcribed in a messenger RNA. Messenger RNA goes to the ribosome. The ribosome is a 3D printer, reads the information, and prints a polymer, a sequence of amino acids, the protein. Protein folds spontaneously, most of the time, sometimes with chaperones that are helpers, but never mind the details. They reach the famous native state I referred to at the beginning of the talk. And that's where the biological function works. And so basically most of rational pharmacology is based on trying to interfere with the biological function of that protein in the native state. In the last several years, there have been attempts, and very interesting and pioneering attempts, to work at upstream, for example, by changing the genome of a person, changing the genetic information of a person so that that protein can no longer be expressed at all. That's an irreversible change, for instance. This is called gene editing. You must have heard of it. Or trying to interfere with the expression of a gene and so far. The idea that Emiliano and I came up with and we patented last year was instead to capitalize on the fact that we were the ones, at the moment, the only ones who could really look at proteins of several hundred amino acids and predict their folding quite accurately, to use this information for drug discovery. So the main idea there was to stop the protein folding process and prevent the protein to reach the native state. Let me be a little bit schematic. You have an unfolded state after you get produced by the ribosome. Before you reach the native state, you have a number of long-lived intermediate states, maybe one, maybe two, depends also on the resolution you look at. And this is the information we are able to disclose from simulations. This is something that was not there until 3 years ago, 4 years ago. Now we have this information and we begin to believe it, most importantly, because of the comparison with the experiments that I mentioned before. So we can design a drug that binds to some of these intermediates and stabilizes to the point that the reaction stops there. Now, if a cell has proteins that are misfolded in some intermediate state and don't move forward to the native state, there's a quality control, a very efficient quality controller, recognizes this protein, tags them, and sends them to the trash can, sends them to a degradation mechanism. And there are several pathways to do that. So in other words, all you need to do is to stop the protein in some intermediate state for some time, and then the cell will take care of it and send it to degradation and will eliminate the protein for you. But if you remove the drug, then you can restore the protein content. So you have a modulable effect, drug-dependent effect, which you don't have if you do gene editing. Because if you do gene editing, once you have changed your background, your DNA, that's for always. So we spent about 3 years in doing all sorts of tests on one of the most difficult proteins to convince that this crazy idea was actually realized. So we collected a huge amount of data, and we worked on prion. Prion is a protein that is involved in a number of prion protein is the prion cellular protein is a protein that normally is healthy and you find in your cells, in the membrane of the cells, especially in the brain. But occasionally they undergo misfolding under certain conditions, and this is the trigger for a number of fatal neurodegenerative diseases, including the mad cow disease that was very popular back in the 90s. It is considered an undruggable protein, no way to interfere with the native structure of that protein. And what this data show was instead that the cellular expression of this protein was modulated by a drug that we discovered through these methods that we developed. It is called pharmacological protein inactivation by targeting intermediate folding, PPI-FIT. So basically, we run simulation of the prion protein, find the intermediate states, scan through a huge database of organic compounds, and try to dock them onto the intermediate states through some typical medicinal chemistry algorithm, find some that are predicted to dock to the intermediate state of the cellular prion protein and stop the folding, and then buy them, put them in the cell, and see if indeed the expression of proteins was decreased. And what this data show is that in fact this is the case. I don't know if you can see the plot on the right, because in my screen it's covered by my face and by the other attendees. But what you see there is a change of expression of concentration of cellular prion protein in the cell as a function of the concentration of the compound that we were throwing into the cell. And you can see there is a clear decrease. In principle, this could be a toxicity effect. We have a number of studies that show that this is not the case. What you see downstairs instead is fluorescent data. You can tag a fluorescent probe to your proteins. Normally, cellular prion protein are expressed and they go to the surface of the cell. So that's why on the left, you see all these nice contours. These are prion proteins, cellular prion proteins attached to the membrane of the cell. But when you expose these cells to the compounds we predicted, then what happens that these contours signaling the border of the cell disappear, and you see that all this signal is concentrated in some kind of very luminous spots, and we believe, because of a number of data we have, that those spots are in fact degradation points where all the misfolded prion proteins get to be digested by the cell. So this first validation was very encouraging. So typically the pipeline of PPI-FIT is the following. First, you have your computer simulations predicting the folding intermediate. Then you have a drug screening medicinal chemistry step where you take, say, 9 million compounds in a database, and then you try to dock them on your intermediate pockets. You find the druggable pockets and you try to dock your compound onto the protein. After you've done that, you go to the lab, you do a primary screening, try to see if you can in fact find some of your predicted molecules do in fact lower the expression levels of your proteins. And after you've done that, if you get to this point, you can move forward and do refinements and what is normally called the hit to lead stage, where you improve the properties, the assimilability, the pharmacokinetics and whatnot, lower the toxicity. And if you passed all this number of developments, then there's mice and humans trial, clinicals, and so on and so forth. Now, as you probably, as Roberto has said at the beginning, we have founded a startup company that follows the upper part of this pipeline. And what I can say is that this company has already found more than one example showing that the prion protein proof of principle was not by chance. We immediately found other examples in which this technology seems to be working. And so when the COVID-19 emergency came up, we thought that was a duty of a startup to stop doing whatever it was doing and try to help out. So we made a collaboration of academics in the upper part, involved in the company, who still work as academics, and the team of the startup company that is downstairs. And we joined forces with INFN because INFN provided huge computational resources. We're talking about 30,000 cores and for a total of 14 million core hours. That was a simulation time that we never had hoped to be able to access. So let me just tell you what was the basic idea of this investigation. I think at this point, most scientists are familiar with how the COVID infection works. You have your virus that approaches the cell. The virus has some external proteins called spike that are distributed on the surface of the virus capsid. And in the surface of the cell, you have receptors which are physiologically there that cause AC2 proteins. Now, what the virus does, it recognizes the AC2 proteins through the spiky protein and uses this as anchor to the surface of the cell, because the AC2 protein is a transmembrane protein. And then once it anchors to the cell, it finds its way to break the membrane and deliver the RNA genetic information into the cytoplasm, providing the infection. So the paradigm we followed in this investigation is that of trying to use PPI fit to find molecules able to lower the expression levels of AC2. If you lower the expression levels of AC2, you have fewer anchors for the virus to come into the cell. That's the paradigm. Whether it will work or not is to be seen. So we did what I promised we would have done with PPI fit, and we used all the resources to fold this protein. But this protein is actually a gigantic protein for simulation standard. It's 600 amino acids, and we folded it in explicit solvent. So this is, for example, an example of a trajectory. It takes some time, but it will illustrate what we do. You start the beginning of the fold, you see, you begin forming what is called secondary structures. These are these alpha helices and beta sheets. And then you fold some kind of intermediate structures, and then all of a sudden you go to this configuration, which is long-lived. This is a nice folding intermediate. Then using some software, we identify druggable pockets in these intermediates. You see, this is the purple area shaded there. And then we started to screen molecules to bind onto this pocket until we find molecules, in principle, that can bind sufficiently strongly to remain attached, and by their steric in volume, preventing the folding. If there wasn't such a molecule, the folding would continue until you reach the final state. Let me say 1 second. Now, here comes the challenge we have to find. Normally, I will say this screening part is done on a database of millions or at least 100,000 of compounds in order to find ones that have the right property. But we don't have this time during the COVID emergency, because developing a drug takes typically 10 years at least, and tens of millions of euros in investments, and there's a long protocol for trials to make sure that you're not killing the patients or anything like this. And of course, we don't have this time here during the COVID emergency. So we decided to do what the drunk man would do, following famous Bethe joke. The drunk man would do, there's this drunk man that has lost his keys home, and he's looking under a street lamp, and his friend says, "Why are you looking for your keys here? Did you lose them here?" "No, but that's the only place I can see." And that's what we did. We look at only through the 9,000 compounds that are already approved as drugs. I mean, they're either on the bench in a pharmacy, or they are in clinical trials. So whatever it takes for these molecules, if we can find one of these molecules, all these 10 years development is basically bypassed, and we can go straight to the last parts of the development. So we screened over these 9,000 molecules and found 35 molecules that were binding in silico. That's already a remarkable effect, because normally we would concentrate, say, on 100 molecules over a database of million proteins, million compounds. Okay, and then we purchased. Isabella purchased these molecules. I must say that most of the real work in this project was done by Isabella and workers, which really are doing something, and they're not trying to profit from the COVID emergency. This, I think, is a remarkable thing I'm proud of. And they have these 35 molecules, and we began to test the first, test them in a row, and we came to a point of testing 14 of them at this point, as we speak. And with our big surprise, we found at least one, maybe two, let's see. We found within the first 14 molecules, we found at least one that displays a prominent effect with a clear dose-response relationship, meaning more dose, more depression of the expression. You see it in the data on the right. and even more importantly, zero toxicity, not zero, but very low toxicity compared to the standard molecules you encounter. So in principle, this is really a smoking gun. And we have to see whether this can work and go forward. This was for us a big, big, big surprise. We were not hoping to be finding a molecule in the small data set we looked at, but apparently, even in the first 14 molecules, we found one. So where we stand on the pipeline, we have this folding simulations, in silico screening, primary screening, all this has been done at this point. There's a slight difference with respect to the previous pipeline, the general pipeline. The next step is to perform a test of the antiviral effect of this molecule. In other words, to test the assumption that if you reduce the number of AC2 proteins on a cell, you actually are able to lower the viral charge of COVID. This is an assumption we have made, and we are working together with the president of INFN to find a way to get appropriate facilities to do this test in a short time, and hope to be able to report in the next few weeks progress in that direction. And of course, if this viral test will pass, then we can bypass all the hit to lead stage and all the necessary step and go directly maybe to some quick animal mouse model and then to clinical trial. So we are making our effort to be as quick as possible. Okay, I'm home in terms of the talk. I'd like to close my presentation with some take-home messages that at least I got from this journey. First of all, really, I mean, I do believe it's one of the examples, certainly not the the most prominent, but clearly one that I lived, that fundamental research matters. Studying QCD, studying vacuum, studying topological fluctuations in strong interactions helped us at least invent a way to try to cure a disease. Maybe it won't work, but it's a way how you go out of the box by fundamental research. This is true in a specific case for theoretical physics, but also major infrastructure you develop for basic research, such as the computer centers that we used, that were kindly given available by INFN, can be repurposed for social emergency. And the second message that I sort of learned out of this is that technological transfer can be also a boost for science. This was sort of something that was not clear in my mind before. I always thought that there's science and then there's industry. And then when you go from science to industry, you go to the dark side of the force. But what this experience has taught me is that if you sort of do good technological transfer, then science and technology and industrial technology can really cooperate together. And industrial research can help boost science research in some situations. Okay, this is the people who have worked. The downstairs people are those from Isabella. And of course, over the last many years, I've had many collaborations, and I list most of them in this slide. And I thank you for your attention.